Professor Fuyu Tamanoi, my wonderful colleague from UCLA, who will speak about signal transduction in cancer and characterization of TSC Rheb mTOR signaling pathway.
Okay, let me start by thanking VNU and especially University of Science for hosting this symposium. And it's been very informative and productive experience, and I'm having wonderful time. So thank you very much. Now, what I'd like to do today is to tell you about signal transduction. So signal transduction is the study of how a cell responds to signals such as growth factors, nutrients, extracellular conditions, or cell-cell interactions or stress conditions. Cells can respond to these and other conditions and then influence the outcome, such as growth and proliferation, or gene expression, or cell division or morphology or motility and others. So cells have a mechanism to kind of respond to outside conditions and then kind of properly behave. And this involves a cascade of protein networks. And in this figure, what I'm showing is example of the RAS signaling pathway, where we know that a different protein is activated sequentially, and that kind of sends the signals from outside all the way to the nucleus. So the study of signal transduction is to understand these players and to understand the process of this protein cascade. Now, the particular signaling pathway that we've been focusing on is called the mTOR signaling pathway, which stands for target of rapamycin. Now, this is a signaling pathway that responds to 3 different signals. One is nutrients by the form of amino acids. So the cells can sense the concentration of amino acids, and if the amino acid is too much in excess, they stop growing. Now, this also responds to growth factors such as insulin or IGF-1. And finally, it also responds to energy conditions by detecting the ratio of AMP and ATP inside the cell. All these signals feed into this signaling cascade that has this mTOR at the central player. Okay? Now, this signaling then responds, the cell responds, and the signal tells the cells to control protein synthesis, and that contributes to the growth of cells, as well as controlling cell cycle progression and also autophagy and others. So I'll try to explain a little bit more as I go along. Now, let me explain why this signaling pathway is very, very important to study. Now, this comes from our study as well as others that looked at different organisms. And it turned out that this signaling pathway is conserved all the way from yeast to humans. In particular, there's fish and yeast that has a complete set of this signaling pathway that we study heavily. But let me just explain what happens if you modulate this signaling pathway. So if you shut down the mTOR signaling pathway in yeast, and this is fish and yeast again, now you can shut it down by inhibiting activator of mTOR called Rheb G protein. What happens is that you see accumulation of small rounded cells. Okay? On the other, and similarly, we did studies with fruit fly Drosophila. And when you shut down this Rheb and shut down the mTOR signaling pathway, what happens is that you get small larvae, as shown here. On the other hand, if you activate this signaling pathway by overexpressing an activator called Rheb, if you overexpress Rheb in head and eyes, then you get you get a fly with a gigantic head, gigantic eye. So you can modulate the growth of cells, and you can modulate the tissue growth by controlling this signaling pathway. Now, the significance of this signaling pathway for human cancer is kind of illustrated here. And that comes from the understanding that a lot of players, many players in the signaling pathways are either oncogenes or tumor suppressors. Okay? So I've shown oncogenes as red here, and PI3K, AKT have a lot of different mutations identified in cancer samples, and these lead to oncogenic transformation. So these are oncogenes. In addition, even though the frequency is low, we have identified oncogenic mutations in the mTOR itself. So these are oncogenic mutations. On the other hand, you have these green players, green proteins such as LKB1, P10, and TSC. These are tumor suppressors. So if you have mutations in these tumor suppressors, that leads to the overactivation of the signaling pathway, and that contributes to the generation of cancer. Now, let me explain a little bit more about these players. As shown here, there are a number of mutations identified in PI3K or AKT in lung cancer, breast cancer, colorectal, and many other cancers. In addition, you see the activation of the signaling pathway or overactivation of the signaling pathway by having overactivation of the receptor that kind of sends signal, too much signal to the signaling pathway. You can actually directly look at the tumor tissue and then look at the activation of this signaling pathway. And when you do that, you find that a large percentage of malignant melanomas have this mTOR pathway overactivated by detecting the phosphorylation of the protein called S6. Now, similarly, if you look at head and neck small cell carcinoma, or renal clear cell carcinoma, or glioblastoma, or myelodysplastic syndromes, you find that a large percentage of them have the mutation, the overactivation of this signal pathway. Now, so the tumor suppressors. So there are 3 tumor suppressors in this signaling pathway, and those are TSC that causes mutations of this TSC gene causes genetic disorder called tuberous sclerosis. And this genetic disorder is characterized by the appearance of benign tumors. Now, the tuberous sclerosis is shown here, and you can see that in kidney, you see these renal cancers called angiomyolipomas that occur in 2/3 of the TS patients here. Now, if you look at the eye, you can see these yellow lesions. Those are called mulberry-like lesion, and these are benign tumors in the retina. So overgrowth is the symptom of this disease. And in addition, patients have neurological symptoms that is due to the change in the brain, and that causes mental retardation, seizures, and autism. Now, similarly, you have this LKB1, and mutation of LKB1 causes Peutz-Jeghers syndrome, and P10 mutation causes Cowden syndrome. So a variety of benign tumors result from this mutation of these genes. So because of all these kind of biological significance of this signaling pathway, we wanted to know the molecular details of this signaling pathway. And I'd like to mention a couple of important points about how this signaling pathway works. Now, at the center, so this slide shows a lot more players in the signaling pathway. as the one I showed in the beginning. So there are many, many players that's identified, and the number is increasing. So it's getting a very, very detailed picture here. But let me just point out that there are two important complexes that play central roles in the signaling pathway, and those are called mTOR complex 1, mTOR1, and mTOR2. Now, both mTOR1 and mTOR2 contain the mTOR protein here, but mTOR1 has a raptor protein associated with mTOR. It also has MLST8 protein associated. mTOR1 signals protein synthesis and growth by phosphorylating the substrate proteins. In addition, very recently, it's been found that this mTOR1 complex affects metabolic regulation, such as biosynthesis of pyrimidine nucleotides. So metabolism is affected by this mTOR1. On the other hand, mTOR2 here, mTOR complex 2, has mTOR, but it has a Victor protein instead of raptor protein. So it has a different protein associated with it. In addition, it has LSD8 and SIM1. This protein controls actin cytoskeleton, and our recent results suggest that filamin A, actin capping protein, is very, very important for this process. And this mTOR2 is involved in motility and cell structure, or the cytoskeleton of the cells. Now, however, I don't have time to go into the study on mTOR2. So in today's talk, I will focus on mTOR1 and tell you more about protein synthesis. Okay? Now, what is mTOR? mTOR is a protein kinase. This is an enzyme that phosphorylates substrate protein and adds phosphate by using ATP. Now, as you know, there are a number of protein kinases, such as serine threonine kinase or tyrosine kinase, and mTOR belongs to serine threonine protein kinase. In addition, all these protein kinases can be kind of viewed as like a protein kinase family, and you can have a family tree, and you can see all these kinases: tyrosine kinase group, protein kinase A, protein kinase C, and many other protein kinases. Now, mTOR is not found here. In fact, mTOR is found in this box here, which shows atypical protein kinases. So it's a very, very unique type of protein kinase. And the reason that this is unique is because it has like a unique structure. And that is shown over here. And this mTOR belongs to this kinase family called PIKK, which is PI3 kinase related kinase family. And in this family, you have mTOR protein. In addition to that, you have other kinases called ATM, ATR, and DNA-dependent protein kinase, which play important role in G2M phase cell cycle progression. All these kinases have this kinase domain at the C-terminal portion of the protein. In addition, there's this fat domain, a fat C domain, that's sandwiching the kinase domain. And then you have heat repeat. So, and this is PI3K that has the kinase domain. Now, this C-terminal half of the mTOR kinase then is responsible for the kinase activity and its regulation. So this is kinase and its regulation. And on the N-terminal side, you have something called heat repeats. It's a repeat of amino acid sequences called heat sequence. And this is shown to, or this is believed to provide a surface for protein-protein interactions. So one simple idea is that you have a variety of proteins interacting here, which will then contribute to the regulation of kinase on the C-terminal region. Recently, crystal structure of this mTOR kinase at its C-terminal half has been reported by Yang et al. Now, the crystal structure only contains this region from here to here. So it's not the whole thing, but it gives some idea about how this kinase works. And you can see that its structure is shown here, fat domain is here, and then FRB is right here, kinase domain is here, and then you have the fat C domain right over here. Now this crystal structure also contains associated protein called LSD8, which binds to the kinase site and regulates what this is needed for the activation of the kinase. Now, I'm giving you a different view here, kind of a slightly different view of the kinase structure. And in this structure, you can see what's going on a little bit more clearly. So what seems to be happening is that the active site of this kinase is right over here. This is where ATP binds. And this is also where the substrate protein binds. And what happens is that the active site is kind of buried inside the protein in a way that the substrates have restricted access to the substrate binding site or the active site. So what seems to be happening is that this mTOR kinase is in a closed conformation so that substrates cannot get access to the active site. And what happens is that you need to have regulation to open up this structure so substrates can go into this active site and catalyze the protein kinase reaction. Now, we as well as others have done an extensive study on mutagenizing this mTOR kinase. And this shows our study using random mutagenesis. And what we find is that we identified a number of single amino acid changes which allow the kinase to become hyperactive or constitutively active. So if you have these mutations, it's always active. So just you are opening up or you are kind of making it active. Now, it turns out that I'm going to skip a lot of details here, but if you look at all these mutations, many of these, majority of them map to this site where the active site is located. So what seems to be happening is that the single amino acid change is opening up this structure so substrate proteins can come in. And the particularly important mutation that we've characterized extensively is called E2419K mutation, which happens at this position. So all these big balls are the mutations that contribute to the hyperactivation of the mTOR kinase. Now, we also identified activating mutations in human cancer genome database. So let me say a few words about that here. So the study on cancer is really kind of accelerated by the development of human cancer genome database. And there are two different databases that we use. One is Sanger Center database called Cosmic Library. Another one is TCGA, the Cancer Genome Atlas. And there's a tremendous database that kind of catalogs mutations found in tissue samples from human cancer patients. And it turns out that at this point, at the last check that we did, there are 8,602 cancer tissue samples that's in this database, and we find mutations in mTOR in 285 cases. And these are single amino acid changes, and we are further characterizing this in detail, but it seems to have different frequency depending on the type of cancer you're talking about. The overall cancer mutation rate is 3%, but it ranges from 0.6 to 13%. And interestingly, endometrium has 13.5% mutation, and large intestine has 7.8% with the mutations. And we're characterizing all these mutations at this point. So there's a huge database that we can use to characterize the mTOR kinase. Now, let me shift the gear a little bit and tell you about how mTOR is activated. As I mentioned, there are three signals that activate this mTOR signaling pathway, and one is nutrient signal that goes through a GTP binding protein called Rheb and Rheb-C. This is a heterodimeric GTP binding proteins leading to mTOR activation, and in this case it's mTOR1. Now, growth factor goes through another GTP binding protein called Rheb GTP binding protein that is needed for mTOR1 activation. And both these signals are needed for the activation of mTOR1. And also energy signal also goes through this Rheb G protein and activates mTOR1. So what is going on here? Let me explain a little bit more in detail here. And as I mentioned, Rheb is a GTP binding protein. That means this protein is active when it's bound to GTP. And then this protein then have the effect on inactive mTOR1, converting it to active mTOR1, and thus mTOR kinase activation of cell growth. Now, this process is controlled by a protein called TSC, which is tuberous sclerosis gene product. And this kind of transmits a signal from growth factors and energy, and that is through the AKT protein. Now, it's also shown recently that this insulin signal also has an effect on TSC to be released from where it functions. So the localization of this TSC and Rheb is very, very important inside the cell. And let me just kind of explain this in the next slide here. And this is taken from Kundi and Wan's Nature Review. Okay, so what happens is that the Rheb is this G protein that is localized to the lysosome. And lysosomal membrane is where all the actions take place. And this Rheb is localized to the lysosomal membrane by its lipid tail. Now, this has TSC and therefore it's inactive. When growth factor signaling comes here, then TSC is kicked out and allowing Rheb to be active, which means it's GTP bound. On the other hand, amino acid signal goes through this another G protein called RAG-A or RAG-C. There are A and B and C and D. So it's either RAG-A and RAG-C or RAG-B and RAG-B, but it's the same thing here. RAG-A is GDP bound and RAG-C is GTP bound, and that is an inactive G protein. But when this is converted to RAG-A GTP bound and RAG-C GDP bound, then what happens is this becomes active, and this recruits mTOR1 to the lysosomal membrane and allows the activation by Rheb G protein. Okay? And this turns out to involve other sets of proteins called regulators as well as BATPase. And this amino acid signal seems to go through this. Okay, so I will kind of speed up a little bit here and tell you a little bit more about how this mTOR1, when activated, recognizes the substrate protein. So the substrate proteins are S6K, that phosphorylated ribosomal protein S6, as well as 4BP1, that phosphorylate that binds to eukaryotic initiation factor 4E, and this is an inhibitory reaction. Phosphorylation 4E-BP1 relieves the inhibitory action on initiation factor 4E, and thus promoting protein synthesis. Now, the important player in this is called a protein called mTOR, which recruits the substrate protein to mTOR1. And so mTOR recognizes 4E-BP1 and then recruits to close to mTOR that phosphorylates this 4E-BP1. protein. Now, we've recently looked at this process in a little bit more detail, and that's shown in the next slide. And in a publication that we just published recently, we found that this 4E-BP1 substrate protein is recognized by raptor protein at its N-terminus, and the central region of the 4E-BP1 is recognized. And we can come up with kind of a computer model to show the interaction between the two proteins. Now let me just kind of explain how we did this in a very brief way here. And what we did is to use a new method called cross-linking mass spectrometry, CXMX. This is a method that allows us to map the interaction between the two proteins. So if you have blue protein and red protein here, you can chemically cross-link the protein, two proteins, and then you break up the protein and you can identify the cross-linked peptides by mass spectrometry. And you can see, as shown here, you can identify a peptide that has a raptor sequence cross-linked to 4E-BP1 sequence. And you get mass spectrum, such as shown here. Now, this is really a powerful method that can allow us to characterize these complexes in much more detail. And as I mentioned here, we analyze the interaction with 4E-BP1. We're looking at the interaction with S6K. We can also look at the intramolecular crosslinks, and then look at the crosslinks between the subunit proteins. And this is one of the pictures that we have, that raptor probably folds up in this way. 4E-BP1, oops, 4E-BP1 is here, and then in close proximity to mTOR. And we're further characterizing these points. But let me just kind of end with the inhibitors. And there are two inhibitors in the signaling pathway that's developed by a number of people. One is called rapamycin, and the other one is ATP analogs. Okay. Now, rapamycin is a natural product that was obtained from a soil sample from Easter Island, Rapa Nui, and this is the secreted product of a microbiological organism. Now, this rapamycin binds to FKBP12 and binds to this FRB domain, whereas kinase, the ATP analogs, inhibit directly or bind directly to the kinase domain. So there are two different modes, and FKBP12 rapamycin complex binds to the FRB domain and further closes the cleft here and restricts the access of the substrates. And the ATP analog binds directly to the active site. And because of these, the drugs, the mTOR inhibitors are in clinical trials, tested in a variety of clinical trials as shown here, and has been shown to have clinical activity with renal cell carcinoma, neuroendocrine tumors of pancreatic origin, and breast cancers, and a variety of clinical trials are ongoing at this point. And finally, I just want to mention that in addition to the mTOR inhibitors, one can use AKT inhibitors, PI3K inhibitors, or dual inhibitors of PI3K mTOR signaling pathway. So I may have gone too much into detail about the signaling pathway, but what we're trying to do is to understand the signaling pathway in details and to understand how the target protein functions, which could provide hints about how the drugs or the inhibitors work, and that could lead to cancer therapy and clinical studies. And as the signal transduction is important, and these are the people that have contributed to the study. So thank you very much.
So this talk is open for questions. Maybe I will start. An ATP analog, one would think, would interact with many proteins.
Right.
Is the side effects Are they an important concern with something like an ATP analog? Because it would interact not only with the intended target, but perhaps other kinases.
That's right. So I think the ATP analogs that inhibit different protein kinases have been studied in detail, and people have pretty good understanding about specificity of each analog. And therefore, I think the concern that it may have side effects is very important. But I think it is possible to have specific effects, and one can now design inhibitors such as one against mutant form of BRAF kinase, and that seems to have clinical activity. So I think there is enough advance to be sure about the specificity, although I think clinical studies are very important. So therefore, the ATP analogs or mTOR inhibitors are now tested extensively in phase 1 clinical studies, and safety has been tested, but we need to look at the results that come out.
Very good.
So when you start to so the activity of the mTOR, that is when the binding pocket of the substrate is open. So so one of those slides you mentioned that you also want to see whether you can see the to understand whether how those when the mutation of the mTOR that they can show the activities. So then you screening with very large libraries, and then at the end you show that there's a certain number of the mutation. But at the end,
I think your points are well taken, because what we need to do is crystallize those mutant proteins and show that, in fact, the idea that the cleft is open is correct. But we haven't done that experiment. What we have done is to introduce these mutations in mammalian mTOR and demonstrate that they behave in a hyperactive, constitutive active way.
Isn't it wonderful that Professor Tamanoi sees crystallography as the road to the truth? We have a question from a student. We should give a prize to a student brave enough to stand up and ask a question. No harm will come of it. Any student like to ask a question to any one of the 3 speakers? It's clear that we need more global mentoring here. Yes, we have a student here who has no danger of Alzheimer's disease.
Well, thank you so much. I think from your work, when you use computational analysis as well as crystallography and relate to, Professor Eisenberg's work, it alludes to the notion that it's a very powerful tool for us to study many biological processes, including drug in research and everything else. However, when we refer to a case like DRAS oncogene, the story is that it has been discovered by Microbiology for almost 40 years, and many technology, including crystallographies and mutation analysis and knockout gene and everything else, to find a candidate drug that can bind the target. At this point, so far, there is no success. Okay. So can you please elaborate and explain how far crystallography, structural biology can go to resolve the problem? Thank you.
Let me mention about RAS signaling pathway, which is another pathway that we studied, but that's going to take another 2 hours, so I didn't do that. But currently, the most powerful method for identifying RAS inhibitors is to use NMR. So you can use high-throughput NMR to identify small molecules that bind to RAS, and then take from there to identify features. And that approach seems to be bearing some fruits, and this is taken by Rheb and Genentech, and they're coming up with possibilities. So I agree that finding RAS inhibitor is very, very difficult, but now I think we're moving into a new type of age when we can now actually find inhibitors. Yeah. Everything takes time, especially.
I would add my minor thoughts to that answer, which would be that the most specific drugs are going to be protein or peptid drugs, because they bind very specifically to receptors. And now private sector, Amgen and other companies have shown that it's possible to find antibodies and other peptid and protein drugs. The problem is delivery, getting them to the proper cell, tissue, and organ. And many laboratories in nanotechnology are working on that problem of delivery. Also, we heard from one of your faculty yesterday about work with natural products. Natural products, such as rapamycin, offer an important route to effective drugs. So it's taken a long time, but I think there's hope.
Good. I am Chancellor of the University of Medicine Pharmacy at UCD, and now the Dean of the Medical School in here. I'm about 20 years in the medical education. And just I want to say there is a very, very excellent progress in biology. Yeah, we have molecular biology, structural biology, and so on. But there is one very big gap on the lab or the result of this and the clinical physics or doctor. And I think this is a big gap because of a long time our curriculum, yeah, for example in Vietnam, there is not enough knowledge about the medical science or the basic medical science. And therefore, I think in the future, when we have the cooperation with UCLA, we need so-called, we have the signal transduction, but I would to say the translation for lab and bedside. This is now very important for the done in pathology. Always there is one sense for the translation then to the laboratory result and the bedside. That's mean for the clinical doctor. This is very important. We need a good doctor with pedagogic method, yeah, to teach what is very, very complicated for doctor. Now for practitioner, it's very, very difficult to understand because a lot of very new knowledge. And I think in the future, when we have good, we can help us to make them more teacher, material teaching, everything, and to make them your result in the laboratory and maybe translation in practice. This is very important because now everything also not for cancer, for cardiology, for diabetology, endocrinology, everything we talk about the signal, transduction, and also for the drug design and drug effect. Therefore, I want to explain this design for the cooperation in the future. Yeah? We need some good teacher, good speaker to make that everything is more easier for the clinical or the practitioner. Thank you.
I totally agree. I think I totally agree with Dr. Fox's comments, and the direction that seems to be coming, or the direction that could incorporate clinical translation, is one that comes from the analysis of clinical samples. And I didn't have time to go into that today. So that's one thing. And having like clinical samples, especially in Vietnamese patients, that is going to be very good. Now, another thing that is important is that there's analysis at the genome-wide level that seems to be ongoing, and that is going to have impact in basic science. And today, I just want to focus on biochemistry and molecular biology, but I totally agree with you that in the future, they have to merge together to lead to multidisciplines.
Yes, you've made a most important point. We all need to tear down the barriers between the different disciplines, and we must construct rapid pipelines that go from the fundamental level to the cellular level to tissues and organisms. And it will only be through enlightened administrators like you that happens. We realize we need your help in doing this, and I think we can provide the teaching help, but we need the administrative help as well. I think we've used up our time, but let me just ask, is there another question for Professor Jonsson Lee? Yes. We need a microphone.
Good morning, Professor. Konnichiwa. My English is not good, so I have some problem when I follow the presentation of Professor about the amino acid of proteins. So I have some questions to confirm that my understanding is right. So the first question, it's not question, in P53 case, I understand that when there are mutations happen, it will have explore the specific peptide, and they will tie together to make amyloid protein. So I wonder whether the amyloid process happen in any proteins. The first question. And
let's do one question at a time. Have you finished the first question?
Yes.
Yes. So I think I understand your first question, which is, does the mutation need to be in a specific peptide, which is the adhesive segment, to form the amyloid, or can it be anywhere within the protein? And if that is your question, my answer is it can be almost anywhere in P53, which is a very unstable protein, as shown by Professor Furch in Cambridge and many others. So all it takes is destabilizing the protein so that it will open a little bit and expose that critical segment. Does that help in answering your question?
Thanks. And so with a given protein, can we predict the decisive peptides? So from that we can establish a way to prevent the disease from the early stage.
So the question is, can we predict the peptide segment which forms the amyloid? And there are different methods for doing that. The method that I illustrated in one slide is a structure-based method, but there are sequence-based methods at all that Professor Lee has used and mentioned in his slide. So there are different methods. And I noticed that various laboratories use both and then look for a coincidence when they do an experiment.
Thanks.
So, and is it reliable? I think so, but others might not.
I believe. Thanks.
Thank you for the question. And we're 6 minutes over, so yes, one more question. Yes.
I would like to ask the professor, and maybe I think that question that when in your group you use structural biology, you saw that the aggregation of the data had two morphology. First one is zipper line barrel. So, what do you have? Could you look into the sequence of the protein or some special thing in your peptide? Could you predict? Could you characterize what is the structure of the? I mean that when you had the sequence, could you specify the factor? Could overlap to become the barrel or the zipper when it's aggregation?
When it's freed from the rest of the protein or part of the rest of the protein? Yeah, so we've had very good success in predicting the segments which form the zipper. Because we predict, we synthesize, they always form the zipper. That works. Then the question is, what's the rest of the protein doing? And we've done some experiments there. And I'll just tell you one. We've worked with the protein ribonuclease. That was the protein that Anfinsen used to show that it can be denatured, and when it's renatured, it forms its native structure with ribonuclease activity. And for that, he received the Nobel Prize in 1972. We like that protein because it never forms fibers. With that protein, we now insert into it one of our zipper-forming segments. When we do that, it forms a fiber. We can insert any of our zipper-forming segments. We've done about 10 of them. Just any one of them forms a fiber. And guess what? The fiber maintains its ribonuclease activity. So that shows, even though it forms a fiber, it has a zipper, but the rest of the protein is in its native form and retains its native activity. So we believe that when fibers form, most of the protein at its native state. I hope that answers your question.
I mean that a little bit assumption question, but I want to ask you, how could you characterize some of the factors of the protein? When it becomes the zipper, when it becomes a barrel, could you specify the factor?
When it becomes a zipper or when it becomes a... Yeah, those are different segments of the protein. So in a protein, there will be zipper segments that can form a fiber, but the toxicity comes from a different segment, the barrel segment. We're less confident in our ability to predict that. Those are the experiments going on in our laboratory at this time. That's an excellent question. Thank you for it. And I think we need to eat lunch. So I believe we'll return at 1:30. Is that correct?